mitochondrial DNA, because the mitochondria only pass from mother to the offspring. The father does not contribute any mitochondria to the zygote. What you're looking at is the maternal lineage of that DNA. Okay, that's the first thing you should realize. But this is the tree they came up with, and the point here is that the first splits in the modern human tree all lead to Africans, people that live in Africa. So the idea is if you were to reconstruct the most recent common ancestor of modern humans, you would reconstruct that individuals having lived in Africa. And unfortunately, because of the maternal inheritance of mitochondrial DNA, somebody thought it would be cute to say, call that individual a mitochondrial Eve, right? Because it had to be some woman that existed about 170, 200,000 years ago that this mitochondrial DNA traces to. That doesn't mean she was the only woman around in that population, but her mitochondria was the one that eventually won. All the others through genetic drift went away, okay? If you do this, you could do the same thing for the Y chromosome, of course. Y chromosomes are paternally inherited, and so you would find by necessity a single male that would have the Y chromosome, and you could call that Y chromosome Adam. But the problem with that is that those two individuals could live tens of thousands of years apart in time. Just a couple other things you can do with, see what I'm doing, how much time. Okay. A few other things you can do with these trees. Here's a genetic tree of different populations of modern humans. You can actually do the same thing with languages. Languages, you probably are familiar with the Romance languages. There's some languages that just seem more similar to one another. Right? So you can actually apply the same phylogenetic techniques that were discussed in lecture to making trees of languages. And the way the data, of course, you can't use DNA sequences or anything, but what they do is they look at certain, what they call cognates, and also words for things such as water, mom, dad, words that you shouldn't expect to change very much over time. And they can actually look at for similarities in these specific words and then make a tree in the same way that you would make trees of DNA sequences or morphological characteristics. But the thing they find out is that these trees, the language tree and the gene trees, are very similar to one to the other, which means that the languages were probably diverging as the populations diverged over time, which is kind of interesting, I think. The last thing one of the last things I want to talk about is Neanderthals. So what are Neanderthals? They're sometimes called archaic modern humans or archaic humans. They're often given the same genus and species name, Homo sapiens. But as opposed to modern humans, which might be referred to as Homo sapiens sapiens, they're referred to as Homo sapiens Neanderthals. They're big, robust individuals. They have brains the same size or larger than modern humans. And this is showing where they lived. They basically had an Asian or a European distribution. Right? These are the points that the red circles show the places at which Neanderthal fossils have been found. And the point to realize is that modern humans, anatomically modern humans, were also in those same areas, which has always led to some speculation about whether what scientists call admixture, or whether you had interbreeding between Neanderthals and anatomically modern humans. Okay? Now, one way you could try to address this question is look for evidence of individuals that look like they might have had a Neanderthal mom and a modern human dad, or vice versa, right? But that would be difficult. I mean, ideally, in order to distinguish whether or not Neanderthals did or did not interbreed with modern humans, you would want DNA sequences. But the problem is Neanderthals are all dead, right? They went extinct about 30,000 years ago. So what can you do? Well, it turns out the fellow on the left, Svante Pääbo, who runs a large research group in Leipzig, Germany, he's become the world's foremost expert on extracting ancient DNA and sequencing it. Okay? And so his lab has developed lots of protocols that allow you to sequence the very small amount of fragmented DNA that you can find in fairly recent fossils, and sequence it in such a way that you can discount the possibility of contamination from the people that are working on that DNA. So he's the fellow. His lab actually was able to sequence the genome of a Neanderthal, which is pretty neat. Who are these other two people, and why am I showing you that? Well, Rasmus Nielsen and Monty Slackner are two scientists on the 4th floor of this building. They are population geneticists. They're these mathematically trained evolutionary biologists who know how to make sense of DNA sequences when it's presented to them. So together, there was this paper that came out a few years ago, basically a draft sequence of the Neanderthal genome. I highlighted in blue all the people that have a UC Berkeley connection. But what they did is they subjected the Neanderthal sequences to mathematical tools that would allow you to detect whether or not admixture had occurred. Okay? And they're specific. So here's a picture of what they do. They basically have to grind out a little bit of the bone to actually extract the DNA. So you have to damage the bone, obviously, to get the DNA out of it. But these are some of the hypotheses they try to distinguish among. So that this is an idealized model of the out-of-Africa hypothesis. You have Homo erectus down here going extinct eventually. We'll discuss about when it may have gone extinct. You have a clade of Neanderthals that branched off from the clade that led to modern humans. And the question is, if there's admixture, A, did it occur? And if so, when did it occur? And their analysis suggested that it was this hypothesis that was the best one, that sometime on the way out of Africa, you had the admixture occur. On the way out of Africa, the admixture occurred. And the reason why they think this is because there's evidence of Neanderthal DNA in all non-African modern humans. Right? So it would have to be the case that the admixture occurred on the way out of Africa. So the idea here is that many of us in this room have 4% of our genome is Neanderthal. Okay, and just a little bit about Homo erectus. Going back to this previous slide, note that Homo erectus is shown going extinct eventually and fairly recently. But the question is, when did it go extinct? Was there any evidence of Homo erectus maybe living to a more recent time? Yeah. And there might be. So this is the island of Flores in Indonesia. And in a cave on this island, it has about 1.8 million people living on it, they found the following. Let's see if I can do this without breaking it, because I get in trouble. Someone might actually let me this, but this is what they found. Yeah. First of all, it's a small individual. So it has many of the characteristics of modern humans, but it's very small. Okay? And so the question is, well, what is this? Is this a modern human, or is it something else? There's two main hypotheses about what this could be. This is Homo florenziensis is what they've been calling it. Okay? If you've heard, it came out, it was discovered around 2003, which is when The Lord of the Rings was coming out. So somebody, of course, had to call it the Hobbit. But the question is this a modern human or something else? And so there's two competing hypotheses. One is that it's a modern human that became dwarfed, so that the population on Flores Island may have become dwarfed. And so this is a phenomenon you see often repeated on islands. It's called insular dwarfism. But basically, when you get an isolated population on these islands, sometimes animals that normally are pretty big on the continental regions over time evolve to a much smaller body size. Okay? That's insular dwarfism. So that's one possibility. This small body size is an example of insular dwarfism in modern humans. The other possibility, which people think might be going on, is maybe this is an isolated population of Homo erectus that just happened to stick around for another 500,000 years on this island. Okay, until finally when it extinct maybe 10,000 years ago. So this could be one of the last strongholds or last holdouts of Homo erectus. That's the other example. But if that's the case, then it would be an example of insular dwarfism in Homo erectus too. Okay. And this is an example just to give you an idea of the size difference between modern humans, the skull on the right, and Homo florenziensis. All right, so a few final thoughts before this lecture and the evolution lectures are over. So I realize that many of you, this is probably your only exposure to evolution, and that we know that statistically most of you are pre-med, which is great. We need really good doctors. But I want to make a point here before I leave, which is pre-med is just a bunch of courses, right? It's just a list of courses that you need to have on your resume. It's not a major, okay? That you can choose any major and still be a candidate for medical school as long as you have those courses. And here, a couple years ago, I went and I asked the curricular advising people here, well, what courses do you need? I wasn't pre-med, so what do you need to take? And it's a horrendously long list of courses, so I'm sympathetic. But there are some of the courses that you need. And I've talked to people that, in medical schools, and the type of people that stand out are like the English major, frankly, that has this list of courses. Somebody went off the beaten path. I don't say you should all go to the English department, but the point is that there's all sorts of different majors that you shouldn't restrict yourself to just certain majors because you think those are pre-med majors. And the last thing I want to make a point of is, UC Berkeley, so what distinguishes UC Berkeley from any other university you could have gone to? So there's, let's face it, the lectures here, I know you probably, I remember this, there's nothing special about the lectures. In fact, I remember some pretty miserable lectures during my time here when I was an undergraduate grill, I guess. And you can get great instruction at all sorts of universities. What really distinguishes UC Berkeley from some of other places you could have considered is the quality of the research. It's the only reason UC Berkeley ever rises as far as it does in any ranking system. And in fact, when you look at ranking systems that only evaluate research, it's usually one or two, right? It's when you factor in other things like how many faculty there are per student that other universities start to trounce us. So my suggestion, I'm not trying to patronize you, but my suggestion is you should take advantage of this resource while you're here, if you can. And one way you can do that is to actually work and do research in laboratories on campus. And there's different ways you can do this. You can go through the UREP program, which is in Durant Hall, just the building over here, the dean's area. Or you can initiate these things yourself. And it's mostly a matter of talking to the professor whose research interests you, right? So what first thing you do is you find out what interests you, and you read that person's at least a few of the papers so that when you go talk to the professor, you're not doing it cold. But then it's just pretty easy. You knock on the professor's door, you greet the professor like I did with Professor Doris Backtrog. There she is. She got over her surprise, and she talked to me about different possible research projects I could do. And she had to be pretty careful because gorillas aren't known for their lab techniques, but she was nice to me. She talked to me, and then you have to be aware of this. Professors can talk and talk and talk much longer than you probably want to, so you have to be aware of that and be prepared for the long haul. But eventually she said, agreed that I could work in her lab. I met the nice people in her lab. They were all very friendly. Here I am working in the lab, the pipette in my hand. But things didn't go as well for me. You want to behave yourself. Sometimes when experiments go wrong, I did what the gorilla would do, but that's not, you have to be patient. Turns out I can't throw things in the lab. So I left the lab, but the point is that you probably won't act like I did in the lab. You probably have a good experience and you'll be able to take advantage of UC Berkeley. So I want to thank the girls, Beth, Lauren, Layla, and Julia, wherever you guys are. And that's it. Go Bears.